A warm welcome to Elimu TV, where you watch and learn. I am your teacher, Madam Chrissy Okot. I welcome you to the sixth lesson of biology, Form 4. We're still on the same topic, genetics 2, and today our subtopic is going to be on inheritance of ABO blood groups. So this is how our lesson is going to be today. We're going to review the previous assignment, going to introduce the blood groups that we have, going also to talk about the inheritance of blood groups, also going to learn about the RHESUS factors and how the RHESUS factors are inherited, and lastly, I'm going to give you an assignment. So let us start by reviewing the assignment. Question 1 was distinguish between complete and incomplete dominance. We discussed this in our previous lesson. So the difference is in complete dominance, the dominant allele will completely mask the recessive one. So it means it is the one which is very powerful. In the example, we have a red flower versus the white flower. So the red flower, the end products or the generation, the offsprings, all of them will be red, meaning that the red flower has overpowered or has masked over the white flower. So in this case, the red flower has shown complete dominance, while in complete dominance, neither of the allele is dominant and it's not going to be shown in either of the offspring. If you have the red flower and the white flower, the offsprings, none of them has the red or the white flower. The products are all the same. They are both pink. So it means none of the allele has been dominantly seen in the offsprings. Question number 2. How are the alleles for incomplete dominance represented? They are represented in a heterozygous form. For example, we have the red and W, both of them. So in complete dominance, neither of the allele is dominant. As in the example, we have a red flower and a white flower. When they are crossed up, when they fertilize, all of them are going to produce, the offspring are going to be pink. So there is no allele which has been seen. So it's shown in a heterozygous form. So there is no flower or there is no allele which is being represented. So we start our lesson by looking at a blood group. So what is a blood group or a blood type? So it can either be called a blood type or a blood group. It is defined as the classification of blood based on the presence or the absence of an inherited antigenetic substance which is found on the surface of the red blood cell. So the blood groups in humans are determined by the presence of an antigen which is found mostly on the surface of the red blood cell. For example, if you have blood group A, it means you have antigen A on the surface of the red blood cell. If you have blood group B, it means you have antigens B on the surface of the red blood cell. If you have blood group AB, it means you have both the antigen A and B which are found on the surface of the red blood cell. While if you have blood group O, it means that you have neither of the antigens which are found on the surface of the red blood cells. So we are talking about the antigens. So let us first look at what an antigen is. So an antigen is any substance that can stimulate the production of the antibodies. Any substance which is used to stimulate the production of an antibody. While an antibody, these are the Y-shaped proteins that are produced by the immune system to help stop intruders from harming the body. We can compare antigen and antibody to a soldier who is on the borderline. The antigen is like the soldier. He is patrolling there to see if there is any invasion of an enemy or an intruder. When he notices that there is an intruder, he will either shoot or arrest that person. So by shooting, those are the antibodies. They are meant to destroy the enemy. So the bullets are like the antibodies. The antigen is like the soldier who ensures that the citizens are very safe, but on seeing the enemy, he will do something about it. So the antigens in our illustration, so these are the proteins. They have a plasma membrane. So your body, if your body thinks there's a foreign organism which is inside, it will produce an antibody. So an antibody, that is the substance which is produced by the body to provide an immune system so that it helps to fight this foreign body which has been inside your body. So we have 4 different types of blood groups. We have A, it signifies that you have A antigens on the surface of the red blood cell. We have blood group B, which means you have the antigen B on the surface. We also have another blood group called AB, which is, it means that you have both the antigen A and antigen B that are found on the surface. Then we have blood group 0 or blood type, which is O. It means that you have no antigens which are on the surface. So we have the main 4 blood groups. You are either A, B, AB or O. So when you look at our blood groups, you can be able to identify the genotype, the antigen which is found in the blood group, and the phenotype. The phenotype is the physical appearance. The genotype is the genetic makeup. So if you have blood group A, you can either have the genotype AA, it means you have antigen A, and your blood group, you say it is blood group A. There's also another alternative of blood group A. It means you have the genotype of A and O. A in this case is the dominant, so your blood group is A. We have a blood group we call B. It means your genotype, the genetic makeup is B and B, and B, they are the dominant, so you have antigen B and your blood group is B. There's also another alternative of B. You can either have the genotype of ABO. It means you have antigen B, which is still the dominant. Then, when you have blood group AB, it means you have both the antigens A and B, which are present. And lastly, we have the blood group O, sometimes we call it the 0. It means you have 0 antigens, you have no antigen. So you have the genotype O and O. So this is how we inherit the blood type. The blood group O, it's made of the recessive genes. The blood group type B, it is made of either homozygous, which is BB, B being the dominant, and BO is the heterozygous. So you have alternative of BB or the BO. Then in blood type A, we have a homozygous state, which is AA. An alternative is AO, which is heterozygous. Remember, homo means one and hetero means different. So if it's homozygous, it means that it's made up of the same genes, and AO, they are made up of different genes. Then we have type AB, which is made of AB. Both of them are codominant. So let us introduce a video. It helps us to explain more on the antigen and the antibodies. Let's have a video that's going to explain more on the 4 blood groups that we have.
I have one other question. Do we all have the same kind of blood? Very good question. Everybody's blood is almost the same, but there is a difference. In antigens, what are antigens? Let me show you one chart. Red blood cells in blood contain proteins. These are called antigens. Oh, the proteins in RBCs are called antigens. Do we all have some antigens? No, not all have the same antigens. There are two types, A and B. We have either blood with A antigens or blood with B antigens, two types of blood. No, actually, there are 4 types of blood. How you said that there are only 2 antigens, A and B? Yes, but there are a few people who don't have any antigens in RBCs. This is called zero antigen blood. It is also called O type. So, few people have A type, few people have B type, and a few don't have any antigen. So, it is called O type blood. And there are others who also have both A and B antigens in their blood, A and B type. These are called AB type. So there are 4 types of blood, A, B, AB, and O.
So that video has been showing how the blood groups are related to the antigens which are found on the red blood cells. So let us look at a cross between individuals of different blood groups and what their offsprings will inherit. For example, we have a man who has a blood type AB and marries a woman who has a heterozygous type B. So if they had 4 children, let us look at their probability of how the children are going to inherit the blood groups. So we're going to use a Punnett square in this case. So in one side you put the genetic makeup of the father, which is AB, and the other side we put the genetic makeup of the mother, who is BO. When you cross over, the first offspring is going to be AB, followed by the second one, who is AO. The third offspring is going to be BB, and the fourth one, BO. So we have 4 different genotypes. We have blood group AB, we have AO, we have BB, and we have BO. So we have 4 different genotypes. One of them is going to have AB, which is codominant, AO, which is homozygous recessive, and BB, which is homozygous, both of them are dominant, and lastly BO, which is a heterozygous. Another cross of a parent between who have O, blood group O, and one who has blood group AB. So the first child is going to have AO, the second child will have BO, the other child will have AO, and the other child will have BO. So this is a 50-50. So 2 children will have blood group AO, so that is just generally blood group A. Then 2 children will also have blood group BO, which is just the same as blood group B. Another example of a cross between a mother who is homozygous for blood group B, it means that the genetic makeup is BB, both of them being dominant, and a father who is heterozygous for the type of AO. So he has genetic makeup of A and O. So if you cross over using a Punnett square, the first offspring is going to have AB, the other child is going to have AB, the third one will either have BO and the fourth one BO. So here the chances are also 50-50. They can either have 2 kids having blood group AB and the other kids having blood group BO, which means the blood group is just blood group B. Another crossover between parents who have both blood group A, dominant, homozygous, dominant, and blood group type B, homozygous, dominant. All the children are going to inherit one gene from the other, which is A, and the other gene from the other, which is B. So it means all of their 4 children are going to have blood group AB, which is codominant. So let us look at the rhesus factors. So human bloods, they contain another antigen, which is also known as a rhesus antigen factor. So among the other antigens, we have a specific one we call a rhesus factor. It is written as or initiated as capital R and a small h. It is also a type of protein which is found on the surface of the red blood cell. Remember, the antigen, all of them are proteins in nature. So if your blood has the protein, it means you are rhesus positive. While if your blood lacks the protein, it means you are rhesus negative. So there are only two options. You either have the rhesus factors or you don't have the rhesus factor. If you have the rhesus factor, it means you are rhesus positive. If you don't have the rhesus factor, it means you are rhesus negative. So these are examples of the rhesus factors that we have. If we go by blood group A, if you have the factor, it means that you are A positive. If you don't have the rhesus factors, you are A negative. Secondly, blood group B. If you have the rhesus factors present in the red blood cell, it means you are B positive. If you don't have, it is B negative. The same applies to blood group AB. It means you have the rhesus factors, and AB negative, it means you don't have them. While still the same applies to blood group O. If you have the rhesus factors, you are O positive. If you don't have, you are O negative. So those are the rhesus factors which are found in the four types of blood. So how do we represent this rhesus factor if you're doing the crossover? So there are two alleles which are involved. So if you have the dominant allele, it is denoted by capital R and a small h. It means you are rhesus positive. While if it is the recessive allele, it is denoted by a small r and a small h. So this is how the genotype is represented. The first genotype, both of them are dominant. So RH and RH, they are both present. It means that you are rhesus positive. The RH is the one which is dominant over the small r and h. So if you have the big R and the small h over the small r and the small h, it means you are rhesus positive. While if you have both of them, the small RH and the RH, it means that the antigen on the red blood cell is absent and you are rhesus negative. So doing a cross of a parent, a father, who is heterozygous, rhesus positive, and a mother who is homozygous, rhesus negative. So when you do the crossover, the RH first will cross with the mother who have both of them, they are RH, they are negative. So it means all of them are RH. So it means two of the offsprings are going to be rhesus positive and two of the offsprings are going to be rhesus negative. So this is a summary of how the rhesus factors go. So we say if you have the rhesus factor, it is the positive one is the one which is dominant, while the negative one is the recessive. It means the dominant one is going to mask itself over the negative factor. So if someone with a rhesus positive blood will either have both of them positive or a positive and a negative, because the positive is dominant over the negative. Then, if you don't know what they are, you have to assume that they are heterozygous. It means they have a positive and a negative. So, for example, if you have a parent who have both of them negative and one of them who has positive and a negative, when you do the crossover, positive and a negative, it means the offspring is going to have the rhesus factor because the positive is the one which is dominant. On the second case, it still applies. The positive is the one which is dominant. It masks the negative rhesus factor. Then the fourth offspring is going to be, both of them are going to be both negative. So we have positive 2 of them and negative 2 of them. So I'm going to introduce a video that is going to help us understand more on the rhesus factors and rhesus compatibility. So that video was showing us about the rhesus incompatibility which is found in human beings. So that has been our lesson for today, and this is your assignment. What type of dominance is portrayed in the inheritance of blood group AB, then in blood group A? Question 2. List all the possible genotypes for blood group A and blood group B. So you can still write to us, you can pose any question, any clarification. Our Facebook page is Elimu TV. Keep those tweets coming. Our Twitter handle is @Elimu_KE. I have been your teacher, Madam Christine Okot. Thank you for watching and see you in the next lesson.